Hello, everybody. I would like to present rather shortly the work on inferring microRNA regulation, which was led by Ofer, and I encourage you also to see the poster on this topic. So let's get going. What are microRNA is all about? MicroRNA are really the bodyguard of cell identity, and they do so by binding directly to the 3' UTR of messenger RNA. And once this is done, there are two events that occurs in any cells, both translational repression and degradation of the mRNA. However, we know that there are, in human at least, quite a few of those genes. We are talking about 2,600 mature microRNA that are apparently the functional units of this action. And it is also known that about 60% of those transcripts are regulated by any of the microRNA in the cells. However, quite surprisingly, this number did not change over 20 years of research from the beginning of the field till now. And we also know that many of the microRNA have actually very poor evidence. We know we are not sure about their activity. So what do we know about microRNA and target pairing? Actually, we know quite a lot. There are hundreds of predicting tools for mRNA and targets, actually too many, because the coherence between them is quite poor. In recent years, there are also hundreds of experimental data coming from CLIP and all kinds of versions of CLIP, but often those are too specific. So the question remains, who might be regulated by microRNA? And this is exactly the project. We ask which gene is a microRNA target. And we do this by a supervised machine learning model in which quite a standard approach, but as in any model of this kind, we need to understand what is the relevant input to the model, and more importantly, or as important, what is a good validation. So the input is really in microRNA comes in many different flavors, molecular layer, the cellular, where and how many in a cell, the network itself, which is quite complex by itself, and some hints from evolution. So let's say something about their molecular level. The molecular level, this is an illustration from one of these tools, of many of those tools. And what you can see is a 3' UTR from here to here. And you can see that there are many, many types of microRNA that might be regulating this. There is a lot of false positive. However, we can see that they are coming in different colors, which means that the affinity or the specificity for each is quite different. So in a cellular level, the situation is as complex, and we can see that from the tissue point of view, we see many microRNA in different tissues, but they are not equal in each tissue. And from the microRNA view, we can see that some microRNA are very abundant, or specific microRNA, very abundant in one tissue, but not in others. So in terms of evolution, we also have important information, but unfortunately, everything that we know, or almost everything, comes from human and a little bit from the mouse, but very few from other organisms. And this is also illustrated here, in which, as we can see, the microRNA that are known are here, and the genes, again, a large number, but we are not sure about more than half of them. So which genes is really a microRNA target, and why do we want to study proteins in order to answer this question? And the reason being is that coding region is under strong selection, and potentially it captures something about the microRNA regulation. So in order to do this study, we took the state-of-the-art of a mere tarbase, which has a lot of information, and we were relying on the strong signal of those that with high evidence or very high level of evidence. We took everything that we know about proteins, including amino acids, the global information on proteins, keywords, cellular localization, organism, drugs, and so on and so forth. And we engineered quite a bit of those information, of those features, to learn everything that we can say about the proteins. Altogether, for each of the model, we built up more than 20 million candidate features coming from all kinds of flavor. And we took the first top 300 features that are selected by the model based on the training data. And those selections done by some entropy base to gain information from those, maximal information from those features. Let's look at some result. We can show that when we talk about the human, we reach our AUC percentage of almost 77%, quite a bit in the mouse, also close to 70%. C. elegans quite nicely, and we have a quite good performance, as we can see here. If we take different models, and I don't have the time to get into it, but we were able to reach a 78% by unifying several of those models. What have we learned about the biology? We learned that some of the features are more important than others. For example, if you have short protein at the cutoff of 349, you are unlikely to be regulated. However, if you have a ubiquitin, you are almost with 25% more likely to be regulated. This is part of the set of features that we learn quite interesting biology. Moreover, we wanted to understand if this biology is related to the function of protein families and to paralogs. In order to understand this, we put all those proteins, in this case of human, and ask what are those proteins that have many paralogs in humans, such as GPCR or RAB proteins that have this number of proteins within the human proteome. When we did this, we find something quite nice, which shows that each family has its own tendency to be regulated. For example, histone basically are not regulated, neither beta defensin. However, the kinases and some other GTPases are highly regulated at a very high level. And this is just the prior level that I showed you before. So can we say something about the paralogs? And to our surprise, we see that the paralogs themselves are not unified by their tendency to be microRNA regulated, but it's only at the level of the family. So another aspect for this approach was we were able to train on organism X and predict on organism Y. And this is shown here when we learn on human and predict on mouse, or we learn on mouse and predict on human. So we have quite a good transfer between organisms. The last point is just to say something about diseases. We were able to show that disease proteins that are important in known disease, such as Parkinson or kidney diseases, are also transferred to other organisms, in this case, the zebrafish. So to wrap up what I showed you that we are able to provide an AUC for human, which is about 75 to 78%. We are generalizing the model across species. We were able to refine this microRNA regulation prediction and to make it more accurate than in the past. And we were able to show that it's really at the level of the protein family rather than the paralogs themselves. And I thank you for listening, and I'm happy to take any question. Thank you very much.